Okay. There we go. All right. So we went through the cycle of sporulation, how a vegetative cell, once it undergoes unfavorable conditions in the environment, it will wall off a duplicated segment of its genome and then prepare that for protection until better times, right? And this is the process of that happening. These endospores, unlike vegetative spores, are metabolically inactive, all right? And we said that that is one of the characteristics of life. You have to be metabolically active. So are spores alive? That's a philosophical question, but I think the answer is yes, because they have the capacity to be metabolically active. Right now, they're in quiescence, but once favorable conditions come back, such as hydration or nutrients or temperature, then it will go back into that vegetative state. Which is interesting because when we think of humans being in vegetative states, we think of them as being in the opposite of a vegetative state for a bacterial cell. Endospores have thick coats and they basically are immortal, right? So we found endospores that have been around for 250 million years, okay? So they can be in that quiescent state for a very long time. And they're impervious to things like disinfectants and boiling. And that is why you have to autoclave it. You have to pressurize the steam at 120 degrees for around 20 minutes. And that is enough to destroy these highly, highly almost indestructible things. Okay. So this is going back to Wednesday's stuff. So sorry, we didn't quite get to this point, but you have to like harken back to Wednesday. We have a bacterium and we realize that it stores phosphate. Which of the components below would we expect that phosphate to be in? What do you guys think?
A.
A, yes, the granules, right? Those are the storage granules, right? Ribosomes, they don't store things, they translate protein. Plasmids, they don't store things, they are genomic material. Lipopolysaccharides, those are on the outside of gram-negative cell membranes, a component of the outer membrane, right? And endospores, those are a protective mechanism. So granules it is. All right, now we are going to shift gears and talk about the shape and arrangement and some degree size of bacteria, okay? There are three major shapes. There's coccus, which is like a ball, spherical shaped bacterial cells. There's bacillus, which is a rod, okay? And then there is spirillum, okay? So coccus is going to be like this. Bacillus is going to be more elongated like that. And then spirillum is like that, okay? And just to make it a little bit more confusing, with bacillus, you can have certain ones that are coccobacillus, so kind of in between cocci and bacilli. Okay, so not quite a sphere, not quite a rod. There's also vibrio, so think of cholera with vibrio. It's gently curved, so it's kind of like a kidney bean shaped rather than an actual rod shape. And spirillum is going to be this spiral shape, right? Helical or comma, twisted, that kind of a thing. And those, out of all the bacteria that we are aware of, and just how diverse these bacteria are, and how many more representatives of bacteria there are compared to eukaryotes, they still conform to these three and a half major shapes, okay? So here is the cocci is going to be mostly spherical, right? Some slight modifications, but for the most part, a sphere, a ball. And this is an image of a Staphylococcus aureus or Staph aureus. Staph means that these circular cocci cells are grouped together in irregular clusters of like grape-like formation. So staph is the arrangement, coccus is the shape. So the other one is strep, right? Streptococcus, and we'll talk about that soon too. But that is going to be a strip, hence strep, a strip or a long chain of cells. The bacillus or rod-like are going to be this one in the middle. And you can see there is some variation of the rod. But for the most part, it's going to look like, let's get to this. For the most part, they look like this one here. That's the common bacillus shape. And Vibrio here, again, that's bacillus, but it's modified as kind of like a kidney bean shape here. What's this stuff here? What are these things?
Flagella?
Yeah, ding, 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 flagella. Okay. Here's spirillum. So as the name implies, or spirillum as the name implies, they're a spiral-shaped helical twisting format. And then another branch of that would be the spirochete. And then they have some like streptomyces, strep meaning strip, that have a branching pattern here. So the strip kind of branches off into other mini strips. Okay, these are less common. Your main shapes when we talk about the ones that are pathogenic to humans at least are going to be cocci and bacilli, the rods and the spheres and the rods. Pleomorphism. We talked about that briefly on Wednesday when we were talking about the mycoplasma, right? Pleomorphism. That looks like it's spelled wrong. I think there should be an I in there. P-L-E-I. Anyway, pleomorphism means different shapes. So some bacterial cells can have more than one shape. And that doesn't necessarily mean it can be rod or sphere, but it's just more like in the size. And so, for instance, this image on the right, these are all different types of the same, or I shouldn't say different types, these are all the same type of bacteria, but they're arranged differently in some cases. So up here you have palisade formation, palisade being just like a fence kind of, so the cells line up like this. Some of them will have these big metachromatic granules, again for storage, right? And depending on how big those get, they will cause the cell to restructure itself and not be uniform all around. So it'll be bigger at the ends that have these granules, and so it gives it kind of a teardrop shape. And that's what you can see right here. Okay, so it's kind of, and then you have these guys where that's almost got that Vibrio pattern to it. Okay, so different types of shapes for one species of bacteria is pleomorphism. And now we get to the arrangements. Okay, this is important, right? You need to know the different arrangements of these cocci and bacilli bacterial cells. Remember, cocci or coccus is spherical, and it can be just one cell, so that's a single. Or if they're in pairs together, that's going to be diplococci. If they are in groups of 4, they're tetrads, right? Tetra, 4. If they're in irregular clusters, like those grape-like clusters, then it's staph. And if they're in chains or long lines, strips, it's strep. And then occasionally these tetrads, if the tetrads continue and kind of stack together, they can form these cube-shaped arrangements called sarcina. The bacilli can also be diplo for 2, so 2 rods stuck together, diplobacilli, chains, streptobacilli, or in those palisades that I showed in the previous slide where you have a bunch of them stacked together in a fence-like formation. Okay. So this is looking at a cocci cell. Here's one. It divides into 2, and now you have diplococci. And then if that keeps on chugging along, you'll get streptococci in that linear strip, okay? And what causes these arrangements is the way in which the cells divide and how they, so when you have one cell divided into another cell, how it orients itself once it's divided, whether it completely disconnects or if it stays connected, and then the way where it goes from there will make it either a chain if it just goes outward, or a cluster if they all kind of tend to migrate toward one another. This is looking at not diplococci, but tetrad formation. So instead of one branching into 2, and then stay and going like that, you have one branching into 2, and then those 2 new cells now, or those 2 cells are branching into 2 new ones side by side, and then all 4 of those divide into another tetrad, and then you get this sarcina, this cube formation. Not as common. The clusters, those are going to be staph, okay? So that's essentially it, right? For the different types of cocci bacteria. You can have single, diplo, tetrad, sarcina if the tetrads start to stack. You can have strepto or strep if it's linear, chain-like, or you can have staphylo if they cluster. Any questions on that? And this should be familiar because, you know, when we talk about different bacterial species, we talk about, we name them based on these arrangements, right? So Staphylococcus or Streptococcus, that gives you information about what kind of arrangement those cells take on. Bacteria, this is just for your information to give you kind of an idea, again, of just how small these guys are. Here's a human hair. And then it dials it down into right here, the scale of things like ragweed pollen and a lymphocyte, a yeast cell. These look like erythrocytes or red blood cells, and then a few bacterial cells to give you an idea of just how big a bacterial cell is in relationship to a T cell, and then how big that is in relationship to a hair, right? So this is at 200x magnification. You can see that hair. And different bacteria are going to have different sizes, right? So the E. coli is going to be about 2 microns, whereas the Staphylococcus is half that size at 1 micron. But that said, you have Ebola virus, which is, per our quiz last week, we know it's a helical form of capsid. And so it can be quite long. It can be just as long as a Staphylococcus bacterial cell at 1.2 microns. However, most viruses are going to be less than 0.2 microns. And in fact, the rhinovirus is really small at 0.03 microns. Okay, so to give you an idea of how these things stack up in terms of size. But you don't need to know this for any quiz or test. You don't need to know that E. coli is 2 microns. Okay, just again, to put it in perspective. Okay, any questions on that, guys? All right. How do we classify prokaryotes, particularly bacteria? We can look at their microscopic morphology. So we can look at whether or not they're strep or staphylo, whether they're cocci or bacillus, whether they have capsules or not, if they're gram positive or gram negative, things like that. We can look at their macroscopic morphology. So when we grow them up on plates, you can look at the colonies that form. And what's the color? Is the colony red? Is it yellow? Is it clear? Is it blue? Is it circular? Is it amorphous? Does it have rough edges? Is it, smooth on the top? We can look at the physiology. Okay, is it a hemorrhagic effect on the host? Does it, is it non-infectious? Is it a beneficial bacterium? Does it not grow in humans at all? Serological analysis, that's going to look at, basically you're taking serum from a patient, from their blood, and you're looking at whether or not there are different antibodies present that correspond to different epitopes on different bacterial cells. So you can classify it in terms of serology. And when we get into E. coli later, we'll talk about how there are different serotypes of E. coli. And so that's going to use serological analysis. And then genetic and molecular analysis. So sequencing, all right, the ribosomal RNA, for instance, looking at it from that degree of resolution. All these different ways are used to classify them and put them into their different bins for further understanding. Here's a phylogenetic tree showing you the relationship of these different bacteria and where they are in relation to archaea, which are the more primitive prokaryotes that kind of gave rise to bacteria and are much more prevalent in very weird hostile conditions. But this will show you kind of the main clustering of these different bacterial types. So cyanobacteria and Bacteroides are going to be more related to each other than the firmicutes and the actinobacteria are. Right? You don't need to memorize this, okay? But this is just showing you another phylogenetic tree, which goes back to chapter 1 when we were talking about what phylogeny is, okay? And it's how we classify different living organisms to see how closely or how distantly related they are to each other. Okay, going deeper into the classification system, we have species, subspecies, we have strain or variety, and then we have type, okay? So a species of bacteria is just, very similar to how we think of other species of other organisms, but it's the bacterial cells that are pretty much the same in terms of their traits and their patterns. But now that we can get the genomes, it's going to be more how closely they are genetically. Okay. A strain is going to be the same species, okay, but are slightly different in their structure or the metabolic process that it undergoes. But a strain is the same species. Okay, so keep that in mind. A type is a subspecies that can show differences in its antigenic makeup. Okay, so again, same species, only it has a different, it elicits a different reaction in the host. Okay, it might have a different degree of pathogenicity, and we'll talk about that again when we get into E. coli and the different types of E. coli, the different serotypes, and how they each elicit different degrees of pathology. Okay, so just be familiar with those differences, because that's what we'll use in order to give reference to different bacterial types, right? Whether it's the same species, the same strain, or the same type. Any questions on that? Okay. Round bacterial cells growing in irregular clusters. What are they? What would be the answer to this one? Without checking the next slide. No cheating.
C.
What?
C.
I said C.
All right. So we know that strep means strip or chain or line. Okay. So these are irregular clusters. So they're going to be staph. Staph is the cluster, strep is the strip. And there's only one staph here, so that makes it easy. But it's also saying it's round bacterial cells, so that should be cocci, not bacilli. Bacilli is rods. So answer is C, staphylococci. And that can be a pretty typical question on maybe a quiz or an exam. Knowing the difference between these arrangements and shapes of bacterial cells. Okay, so what are the weird ones, right? There are plenty of bacteria. In fact, the majority of bacteria are free living and do not cause harm to us or things dear to us. There are bacteria that are photosynthetic, right? Bacteria was the first life form to embrace the sun and harvest the sun's power to get nutrients, right? Cyanobacteria, those are the blue-green algae. There's also green and purple sulfur bacteria. Those utilize sulfur, so they can be found at these hydrothermal vents, and they obtain nutrients by breaking down the sulfur. Gliding and fruiting bacteria. These are going to be, again, very unusual, atypical bacteria that nonetheless deserve some kind of spotlight in microbiology as we're talking about the different flavors of bacterial life. Cyanobacteria, that's going to be gram-negative. So gram-negative, they have a thick or a thin peptidoglycan?
Thin.
Thin, yes. They have extensive thylakoids, which if you guys have had biology, I'm sure they've talked about photosynthesis, and those are these stacked structures in plants and in these blue-green algae that allow photosynthesis to occur. That's the site where the chlorophyll is present. And so that's what's shown here. Green and purple sulfur bacteria are also photosynthetic, and they have different pigments though, and so they can be expressed as purple in some cases. And again, they are often found in places with sulfur, and so they can utilize that and they can survive in those environments. Gliding and fruiting bacteria, also gram negative. They can glide over surfaces. And what do you think is one way that they could do that? How could they glide over a surface? Anybody know? Oh, sorry. Maybe a glycocalyx slime layer. Remember, that's what the slime layer is for, is to help glide across surfaces. So they probably use that. And what happens is they form this aggregation. So these vegetative cells will divide, but then they'll kind of all cluster together and continue to to form this community that eventually gets a three-dimensional morphology and starts to grow into this glob of a sphere, which is called the fruiting body. And then that forms a myxospore, and then it opens up and releases the vegetative cells to then go and do it over again. Okay. Not super common, but nonetheless worth mentioning. Okay, let's talk about the unusual ones that do have a medical significance, though. Okay, so the rickettsiosis is one of those examples. It's an obligate intracellular parasite, so that means it has to go inside the cell of a host in order to carry on its life cycle. These are going to be gram negative, very small. Most of them are pathogenic. And without a host, they cannot multiply and divide and survive. They need to have the host. And so this can be an example of Rocky Mountain spotted fever, which one of you originally were going to talk about. So it's a pretty gnarly disease, and these are pretty unusual bacteria. Another unusual form is going to be the ones that cause chlamydia. Those are also small. These are also obligate intracellular parasites, so they have to be inside a cell to survive. They're not transmitted by arthropods, okay? So it's not transmitted via a vector. It's usually transmitted sexually, okay? And if it reaches the lungs, then you can have a form of pneumonia that's from chlamydia, right? So transmitted from human to human. Any other questions so far? Okay. So archaea, the other prokaryotic life form, is going to be that third domain in the overall classification of life, right? Prokaryotes, eukaryotes, and then archaea. They are actually more closely related to eukarya than to bacteria. And that was that question. I think that was one of the concept checks we did in the first week. And if you look at that, where's that tree of life? Sorry, going back here. If you look at the branching here, they are more closely related to things like these protozoa and these algae than they are to bacteria. But that's only on a genetic standpoint. Really, there seem to be more, they're more closely, they resemble bacteria more than they do a protozoan, but their genetic components are more closely to eukaryotes. Okay. And what makes them unique is just their structural components. As I said, they're able to endure environments that are inhospitable to most other things. Here is a table that you don't need to memorize, but does give you some of the, shows you how these archaea stack up, right? So even though they're prokaryotes like bacteria, they don't have a peptidoglycan in their cell wall. Neither do eukaryotes. But like bacteria, they, for the most part, lack sterols in the membrane, which eukaryotes do have. And remember, sterols are these alcohols that form cholesterol and things like that. There's some exceptions because, as we mentioned, the mycoplasma have sterols that make up their membrane, and that's what allows them to be pleomorphic in all different types of sizes, right? Okay. So, so there are similarities with bacteria. There are similarities with eukaryotes, and so that's why archaea are somewhere in between. It's like they gave rise to bacteria. Some of the archaea gave rise to bacteria. Other archaea gave rise to eukaryotes. These are going to be your extremophiles, okay? Extremophiles means they like extreme conditions. And like is maybe kind of an anthropomorphic term. More specifically, they are able to subsist and exist in extreme conditions, okay? So they are tolerant to high temperatures, to high salinity, to low pH, to pressurized systems, okay? These are going to be the organisms that produce methane, called methanogens, because they produce methane as a byproduct of their metabolism. Hyperthermophiles. Anybody know what a hyperthermophile is? What do you think that is? Thermo?
Someone that loves high heat.
Yeah. Thermophile. I don't think, maybe I might have mentioned this, I don't remember. But PCR, polymerase chain reaction, is carried out by taking a small amount of DNA and then heating it up to break apart the double strands, and then cooling it down a little bit so that these primers, these little short segments of nucleotides, bind back to the complementary sequence on that single strand of DNA. And then a polymerase, which then attaches and sees that primer and then starts transcribing a new strand of DNA onto that initial template. And why do we do PCR? We do it so that we can take a small amount of DNA and turn it into a large amount of DNA. Okay, and that's been a godsend in forensic science, right? So DNA evidence, it used to be you could collect some DNA from a crime scene, but you couldn't expand it enough to then start sequencing it and doing tests on it, right? So now that's changed thanks to PCR. We can also modify DNA by introducing certain mutations or correcting mutations. So anyway, where I'm getting at is when PCR first came out, you had to have these different water baths essentially, or if you had a machine, this person running the PCR after each cycle where it went up to 90 degrees and the DNA fell apart from each other. And then it cooled down to the primers, and then it rose back up to about 70 degrees to start transcription. after each round of that, you would have to add new polymerase, because a polymerase is an enzyme and the human body doesn't get up to 90 degrees Celsius, right? If it does, you're done. So that would denature the polymerase each time the temperature got up to 90 degrees to separate the double strand. And scientists are lazy. That's how technology flourishes. We come up with innovation so that we don't have to do things. We allow machines to do it. Or in this case, instead of having to be there at that PCR machine, adding polymerase each cycle, someone finally found a solution. And what do you think that solution was? Any guesses?
High temperature polymerase.
Yes, right? So it's called Taq polymerase. So if you do PCR, you go get all your reagents, and one of them is going to be a Taq polymerase. And T is for thermos, and AQ is for aquaticus. And what it is it's a polymerase enzyme from a thermophile, Thermus aquaticus. And so all that means is that that polymerase can withstand that high temperature. And now you don't have to add new polymerase each time because it doesn't denature at 90 degrees. And so it can continue to do its job throughout the entire PCR cycling, which is usually 35 or more cycles, depending on the application, right? So rather than have to stand there and every like 30 times add new polymerase, once and you're done. Okay, that's enough of that. But this is kind of it, really. This is the last part of the chapter, and it's going to just go into the different taxonomy based on this guy, Berge. He came up with it. He says a manual, and it just breaks it down into different volumes of relativity. And I'm not going to make you guys know any of this material. I'm going to go over it because we still have time. We still have half an hour. But this is basically here just to give you an idea of how things are classified and where we group certain bacteria. So if you want to glaze over right now, this is the one, probably the only time I'll give you permission to do so. If you want to leave and work on your proposal outline, by all means, you can drop off, because like I said, this part isn't going to be on the midterm or the quiz or anything. Okay. So if you do sign off, have a great weekend. I'll see you on Monday. If you have any questions in the meantime, send me a message. But then now I'm going to get into this taxonomy and go through it. You're welcome to stay. You're welcome to leave. It's up to you. Okay. So there are 5 major groups within this taxonomy system. 2 domains, so the archaea and the bacteria. It doesn't include the eukaryotes, because this is a manual on bacteriology, and we just lump in archaea with that. So these are the 5 different sections. Now there are going to be 5 volumes, but 6 different categories, because one volume combines both the archaea and the bacteria of particular flavors. Okay. So volume 1 is the archaea, and we've already talked about these. These are the unusual characters. They're thermophiles. It can be acidophiles. What do you think an acidophile is? Well, that's something that thrives in a low pH condition, high pressure. They can be anaerobic, meaning they don't require oxygen. Halophiles. Anybody know what a halophile is?
Isn't it salt?
Halophile. No. Any other guesses?
Isn't it salt? Something with salt?
Yes. So a halophile is going to like salinity. So what do you think an example of that would be? Elkie.
Um, ooh, what is it? Monolake, maybe? Some
Yeah.
kind of bacteria in monolake for archaea.
Absolutely. Absolutely. And so that's why I believe I could be wrong, but I think Mono Lake is called mono because it's pretty much devoid of life, right? Am I correct in that?
Oh, I don't know, but that sounds mono
Yeah.
being one
Right.
and one lake.
Right. And I think they found I think they found I think they found some nematodes in there. But for the most part, yeah, it's going to be so salinized that the salt um, isn't conducive for life for most things, except for these halophiles. Um, that's all you really need to know. I mean, you don't need to know any of this classification thing, but I would know a little bit about archaea. Like, are they more closely related to eukaryotes or bacteria? Eukaryotes. What are some characteristics about them? They're thermophiles. They could be halophiles. They could be acidophiles, right? That kind of thing. Then the other aspect is looking at the phylum. And again, you don't really need to know these particular phyla, but it does have some information in here. Like, for instance, methanogen. What is a methanogen? It is an organism that produces methane. What is a halophile? That's going to be an organism that can tolerate high salt. Thermophile, high temperature, that kind of thing. Domain 1 is then parsed out into bacteria. And so these are going to be some of the oldest bacteria known. Okay. And so the reason they're in volume 1 is because they are most like the archaea species. Okay. So these are going to be, again, halophiles. They're going to be thermophiles. They're going to be able to be photosynthetic. They can hang out and metabolize sulfur, these extreme conditions, just like archaea, which is why they're grouped into the same volume. Here's some examples. Okay, you don't need to know these examples. It's just there for your information. But think of the deep sea hydrothermal vents that come out. There's going to be all kinds of strange extremophiles that live there, or even the sulfur vents and the hydrothermal vents in places like Yellowstone. It doesn't have to be a deep sea vent. There's all kinds of different bacteria that are lurking there, that have been there because they can survive it. So some more phyla of the bacteria in volume 1, cyanobacteria, again, photosynthetic. Some of these live in fresh water that's very hot. So again, like your hot springs and pools in Yellowstone, for instance. And then that brings to volume 2. This, along with 3 and 4 and 5, this is where we have the bacterial characters that are most relevant to humans in terms of the medical impact they have. Okay. So volume 2, 3, and 4 and 5 are going to be the ones that are particularly relevant to humans from a disease perspective. So proteobacteria, gram negative. Now, as we've seen, there's plenty of proteobacteria that live in our gut that make up our gut microflora. Okay, so not all of them are bad, but some of them can be, such as E. coli and Salmonella and Vibrio cholera. These are all proteobacteria, that phylum. And here's some examples of those. So Rickettsia, which is the agent of Rocky Mountain spotted fever again, E. coli, the O157, that is a particular type or serotype. And then there can still be thermophiles, right? Volume 3 is the firmicutes. And so again, these are ones that are living and healthy microflora, right? And they provide benefits to us. They aren't pathogenic, but there are some that can be. Some can form endospores. And that can be things like C. diff. There are different shapes that can be Staphylococcus or Streptococcus. So it's a variety of arrangements. And then it also includes the mycoplasmas that have that sterile cell membrane instead of the typical cell wall. Okay. And the reason those are lumped in with the firmicutes is because of, again, not morphology, but genetic relationship. And here are some images of that. So Bacillus anthrax, that's going to be a firmicute. Streptococcus pneumonidae, which causes pneumonia, that's going to be a firmicute. That said, there are plenty of firmicutes in the gut that aren't pathogenic. Actinobacteria. So again, another phylum that we find in the gut that is healthy and good for us, but then some that are pathogenic. Mycobacterium. So that's going to be your tuberculosis, right? So some good, some bad. All gram-positive. And then it says it includes a high GC content, like over 50%. Do you guys know what that is? Any guesses what GC content could be?
Glycocalyx, maybe?
Great guess. That's what I would think. This is actually referencing nucleotides. So guanine and cytosine. So AGCT. Those four nucleotides have a certain distribution in a genome. And when you look at GC content, you're looking at the stretches of the sequence that is only G's and C's with no A's or T's interdispersed. And so if over 50% of the genome has a GC content, that's going to give it a unique genetic signature, different than most things, which has a more evenly distributed array of nucleotides. And here's some images of that. Okay, so again, tuberculosis, streptomyces, and streptomycin, right, is an antibiotic. So these are species that can be beneficial to us in that they help us produce antibiotics that we use, that they use against microbes that try to invade their space, right? So just as they use them to keep other microbes at bay, we can exploit that and use it to keep microbes in us at bay. Volume 5 is going to be really kind of a mixture of all of them, of all the phyla we've talked about that are gram negative. Okay. And so this is kind of a gamish, if you will, of different phyla representatives. So that's going to include things like chlamydia, spirochetes, right? We haven't really talked much about spirochetes, But one of the spirochetes that is particularly unpleasant is going to be that which causes syphilis. So, and that's an image, I believe, of, yeah, that's Treponema pallidum, which is a spirochete. And so, so those are some examples. Planktomycetes are another kind of unusual one. And so what's unique about this is that it does have a membrane around their DNA and special compartments enclosed in membranes. Ooh. And so what have we been saying is the major difference between eukaryotes and prokaryotes? No membranes, okay? But what have I been saying all along? Everything I say has an exception, right? And so this is the exception. These are really interesting bacterial organisms that are believed to maybe be ones that gave rise to eukaryotes. Now, that's a hard sell because, like I said, if you look at the phylogenetic tree, it seems like eukaryotes branched off from archaea. And so, you know, you have to then decide, well, which is it, right? Bacteroidetes, again, that is a very common gut microbe that is not particularly bad. It can be a normal resident. They're gram-negative anaerobic rods. So if you look at this picture here, what do you think this is? It's bacillus. What would we call that, though, based on the arrangement? Does it look like a strip? We would call it streptobacillus, right? Because they're in these long chains of rods. They're anaerobic, so they don't need to have oxygen, which is good if you're going to be living in an intestinal tract, which doesn't have a high degree of available oxygen ready. But then, you know, some of them can be bad. While some of them are good, plenty of them are good, some can lead to infection. Okay. And so that's that, guys. That is the chapter 4 in a nutshell. Again, the Bergman or the Berge manual classification system is not going to be tested, but I just gave it to you because I think it's good if you're interested in this. It helps you dig into it a little bit more and see how things are grouped, how they're related, how they're not related. And we're going to delve into the medically important bacterial species in much more detail in later chapters in the quarter. We'll be talking about all the gram-positive bacteria that cause disease in humans, and then we'll go into gram-negative. And so we'll have much more resolution than this Berge classification. So I'm not going to hold you to it just yet. Okay. Any questions on that? Okay. We've got about 10 or 15 minutes. So if you have any questions or comments on anything related or unrelated to this class, now is the opportunity to bring it up. If you saw something cool in the news, I'm all ears. Or if you want to just hop off, then that's fine too. So I'll leave it up to you guys to direct the next 10 or 15 minutes.
One cool thing when we talked about Mono Lake just recently, I had to go check real quick, and they said that they found an organism that uses arsenic, and that's extremely rare.
Yeah. Was that a nematode?
You know, I don't know. I quickly got back onto the lecture, but I had to go look real quick. Let's see if it's still on my.
Yeah.
Mono Lake, yeah. First known life form that uses arsenic to make its DNA proteins. It's in National Geographic. I'd have to open that up. It's a new bacteria.
It's a bacteria. Okay.
Yeah.
Yeah. Yeah.
And when is this?
That's pretty cool.
2012.
2012. Okay.
Huh.
Yeah. So I think since then they found a unique nematode in that lake as well that I thought was able to at least process arsenic in some way, that it wasn't
Cool.
killed by it. And so yeah, so there you have it. These are going to be, that's an example of an extremophile, right? And I don't know
Yeah.
what you would call that, an arsenic file or something, but yeah. Rare creatures live, and that's again why we say that microbes are ubiquitous. They're everywhere, right? They're in the places that most living things disdain, yet they have eked out a niche there.
Mhm.
And so,
That leads me to a question I remembered I had. Are there right now, have they discovered bacteria, or are they trying to develop bacteria that can actually exist off of nuclear waste materials?
I don't think they've, I don't think they've, I don't know, to be honest.
Okay.
Not to my knowledge, there's anything in existence yet. But that would be interesting. If they could do that, that would be,
Yeah.
I don't know how they would do that, but yeah, that would be very interesting to see if that could somehow break down and speed up the rate of decay of radioactive samples
Right.
without
Yeah.
without having it damage their own genetic material in the process.
Right.
Yeah.
Right. Okay, cool. All right, awesome. Thank you.
Yeah, you're welcome. Bye.
Hey, Carl.
Yeah.
I was wondering if you got my email about November 2nd.
Oh, yes, I did. So let's see. We are going to be covering, yeah, there'll be a journal club and stuff like that. So yeah, we'll find a way for you to make up for that. Especially
Okay.
thanks for sending the doctor's note. I appreciate that.
Yeah, of
Yeah,
course.
yeah.
And then like on Wednesday, like even though like there's a possibility, just depending on like how I feel after the surgery,